When scientists say solve a structure, what does that even mean? Basically, it's figuring out the way that a molecule's atoms are arranged in 3 dimensions, kind of like its architectural layout. So if we think about our protein, we have all of this primary sequence of amino acids all linked together, and we can know the actual atomic composition, but we don't know how those atoms are actually arranged in space. And that's what solving a structure tries to do. It uses techniques like X-ray crystallography and cryo-EM and NMR, as well as kind of like modeling techniques like AlphaFold, in order to figure out the molecular arrangements of molecules like proteins and even complexes of proteins and nucleic acids and all these cool things. Basically, that's what we mean by solve a structure. And anybody in the world, including you, can then go access these structures and learn about these awesome molecules, or maybe just marvel at them, and really begin to get the beauty of structural biology, which tries to connect form and function and figure out the relationship between what a molecule looks like and what a molecule can do. So remember our primary sequence, that's just our sequence of amino acids. And so basically we can draw a long chain. We can say, okay, well, we got all these peptide bonds and stuff like this. We know which atom is connected to which atom, but that's not going to tell us how they're actually going to be folded up in 3D. And so we want to go and say, okay, well, how are they folded up in 3D? What is our secondary structure and our tertiary structure, and maybe even our quaternary structure? We can use this using structural biology methods. So the main structural biology techniques we're talking about X-ray crystallography. This has kind of been the conventional workhorse of structural biology. Lately, there's been a revolution in cryogenic electron microscopy, or cryo-EM. It's been like this revolution. There's been a bunch of advancements in the technology. Originally, it was only good for like really big things, but they're starting to get down where it's also being able to be used for smaller protein structures, as well as structures of complexes and things like this. So we'll go into a little more detail, but still just at the very lowest level. There's lots more detail, but basically we'll take a look at these different techniques. But first, there's also a couple more. One is nuclear magnetic resonance, abbreviated NMR, which is good for small, small flexible things. And then there's going to be kind of computational methods. So you're going to see things like AlphaFold, Rosetta Fold, etc. And so we're talking about like AI, machine learning, all this various things. There's also like molecular dynamics and things like this. So lots of different techniques that you can do on the computational side. But then you have these, which are going to be experimentally determined. And so that's going to be like actual wet work. Okay, so those two main ones that you'll see these days are going to be X-ray crystallography and single particle cryo-EM. So we're distinguishing single particle. When we talk about a particle, basically that's just like, that could be your protein, that could be a protein complex. It's the thing that you're trying to get a look at. There's also methods for electron microscopy that are not single particle, that are basically looking at like tomography, or basically like slices of cells, things like that. There's some really cool stuff going on in those fields, but we're going to be focusing mostly on the single particle. The basic idea is that with X-ray crystallography, what you're doing is you take a protein and you get it to kind of crystallize. And this is a lot harder than it sounds. And basically what it means though is that all the protein copies are going to kind of line themselves up in an orderly array. So if you think about like a salt and a salt lattice, how everything's kind of interconnected in the same orientation throughout, you get the same thing happening, but with a protein. Then what you're going to do is you're going to shoot X-rays at it, and those X-rays are going to interact with the electrons in the protein or whatever other molecule you're working with. And then those interactive waves, they're kind of going to get scattered. Those scattered waves are going to interfere with one another. Think of dropping a golf ball into a swimming pool and all those like little ripple waves, how they interact with one another. The same thing is happening here, but we're dealing with X-rays. And then they're going to go hit a detector. When they hit a detector, they're going to get make a pattern of spots called the diffraction pattern. And then scientists can work backwards from this pattern in order to figure out the structure of the molecule. So I did some of this in my grad school days, and it can be really cool, but it can also be really frustrating when your protein doesn't want to crystallize for you. So you have to try out lots and lots of different conditions. What about cryo-EM? Well, with cryo-EM here, basically, you're kind of, it's more like taking pictures, but you really fuzzy ones. And so you can't just take a picture of a single molecule. That wouldn't be very helpful. It'd just be like a fuzzy thing. So instead, what you're going to do is you're going to take lots and lots of pictures of the molecule. And what's nice about cryo-EM is you don't need to get it to like crystallize. You are going to freeze it in place, but you're not making it kind of freeze. You're basically freezing it by literally freezing it, or at least vitrifying it. So basically, you get it super cooled in a really thin layer of water that serves as like this glass. And then what's going to happen is that your proteins are kind of going to get stuck in place wherever they are. So they're going to be moving around randomly, and they're going to be kind of like, so you might see some like laying down or standing up or things like this. And then what's going to happen is that they're just going to kind of get stuck in the position that they were in, rather than crystallography, where they get stuck in that position that's going to be the same throughout the crystal. Now what's going to happen is you're going to take lots and lots of pictures of these using electron beams. And so because they're so tiny, we're going to be using electron waves instead of light waves, because light waves aren't going to be small enough. So we're going to use these electron light waves, and we're going to focus them by electromagnetic lenses, and then they're going to hit a detector. And then there's a bunch of computational stuff that will give us those fuzzy pictures. We get fuzzy pictures of the protein in all sorts of different orientations, and we kind of average them together. So we'll say, okay, well, those ones all look like they're laying down, and these ones all look like they're standing up. And so let's put these together. And then, okay, well, now we have the same thing in different positions, so we can average those all together. We can use them to make a 3D model, similar to like a CAT scan, where they take different slices and then they kind of like work backwards to piece the 3D thing together. And then you're able to figure out what the structure is based on averaging together all those fuzzy images. You can also, with cryo-EM, because they're kind of things can be in different conformations as well. And sometimes you can see those too. So for example, you might see someone sitting instead of standing or laying. And when they're sitting, well, then they're in a different shape. And so if you can isolate, like, okay, well, we have ones that are sitting, and we'll isolate those. And then we have ones where you can see the back of the person sitting, ones so you can see the front of the person sitting. We can average all of those together. So basically with cryo-EM, there's ways to see different orientations of molecules, like different conformations of molecules, as well as just being able to average together all of those different kind of like orientations of the same conformation. Bottom line, you need a lot of data. You average them all together and you can get a structure. But it's not as easy as it sounds. And with any of these techniques, when we're talking about cryo-EM, when we're talking about crystallography, what we're getting is actually we don't get the positions of the atoms directly. Instead, we just get the evidence that they were there. So when we're dealing with crystallography, we're dealing with kind of like evidence of where the electrons were. And so when we're dealing with cryo-EM, then we're dealing with technically we're dealing with like Coulomb potential. Anyway, you get this like fuzzy looking kind of map thing, and then the scientists have to go and kind of model things into those maps. So when we go and we look at structures, what we're looking at is actually a structural model where they're modeling in the positions of the atoms into that data. And so we'll look at how we can actually go and explore these models ourselves. The higher the resolution of the data, kind of like the less fuzzy it is, and the crisper the maps are going to be, the easier it is to fit in the model. So when we talk about resolution, a high resolution is going to have little numbers. So
High resolution, little numbers. Low resolution, high numbers. High resolution is going to be good. Low resolution is going to be bad, which you can probably guess based on if you think about a high-res TV versus a low-res TV, you want that high-res, right? Well, resolution is referring to how close two things can be while you can still kind of like tell the two different things. And so the smaller the number, the closer things can be where you can still tell them apart. So it's like if you have two dots far away, can you tell that they're two dots or do they just look like one dot? And basically the better your eyesight, the closer those dots can be and you can still see that they're two different things. So similarly, the higher the resolution of our data, the easier we can kind of say, okay, well, yes, there's an atom here, there's an atom here, there's an atom here, versus, oh, there's some sort of sausagey thing we'll have to fit things into. So that's what you'll see when we talk about resolution, and we're typically dealing in terms of angstrom, which remember is a 0.1 of nanometers. What's really great about structural biology for the outsider is that all of the structures and the experimental data behind them, like their maps and stuff, have to be deposited so that anybody can go and look at them. And they get deposited into this thing called the Protein Data Bank, or the PDB. You can find all sorts of information about how the scientists solved it. You can actually go and explore the structure. You can check out different features about it. And so we'll be looking at the PDB, and they have some great educational resources. So check out PDB 101 for more info on it, on how to use the PDB, as well as more info on structural biology. Another technique is nuclear magnetic resonance. So this is going to be good for small, flexible. What happens with crystallography and with cryo-EM is that basically there can be regions of the protein that don't resolve. So you don't actually get maps for those regions. The part of the protein is actually there, but the data is too fuzzy. So you can kind of think of it all averaging out. It's all blurring each other out, canceling each other out. And so you're not going to see those regions that are flexible. But with NMR here, it can actually use those flexible regions, but it only can deal with small things. But they're going to be like randomly moving around, and what you actually get is some sort of ensemble. And so you can check out the video if you want to learn more about this. The PDB has a little video explaining it, but we don't need to worry about NMR for the purposes of our class. The other thing you'll hear a lot about is like these AI and machine learning, various ways to predict structures. And you'll actually see that in UniProt, so it's going to be the protein database we'll look at. Basically there you can actually see predicted structures as well. So there are limitations, like if you want to look at complexes, if you want to look at regions of proteins that don't have structures that are like known structures kind of, or regions that are kind of more flexible. If you want to look at modifications, there's various limitations to these prediction algorithms, but they often can be helpful for structural biologists to kind of like get an idea of what a protein looks like and maybe even use it to help make their models. So there's various things like this that you'll probably hear about lots.
So if you want information about a protein, you want to go to UniProt. UniProt will also give you links to PDB, where the PDB, that's where you're going to go and find structures or these structural models. But say you want to find a structural model corresponding to a protein you're interested in. What you can do is just search for the protein in UniProt. So let me go and maybe I want to look for human mitochondrial malate dehydrogenase. And I could actually have just gotten it from my cart, because you can add things to your UniProt cart. Just click on it, add it to your cart, and voila, it's here for you to revisit later. But this is what I want to look at. And so I'm going to look at the entry for this UniProt entry for malate dehydrogenase, the human mitochondrial version. If I scroll down or I just click on this structure tab, well, now it's going to show me all of the kind of like information about all of the PDB entries for this protein. So if you see a PDB entry, the PDB entries correspond to experimentally solved structures, and it'll even tell you the method used to solve it. So all of these were solved with XRay crystallography. And here you can see like the resolution of the structures, where the higher the resolution, the better, but a higher resolution corresponds to lower numbers. Here you can see what positions the structure contains. It's always good to go when you're in this structure tab and make sure you're looking at a structure for like the full length or close to the full length or something, or at least know what you're looking at. So it kind of defaults to going in like alphabetical order, I think. And so what happens is that you could be looking at just like a small part of a protein, because scientists often, especially if the protein is really big, maybe it's got really big floppy areas, it's hard to get the full structure of the protein. And you're going to therefore just get a little bit of the protein. And so a portion of it. And so you see the positions would be smaller. And so you might be looking at something and you're like, that doesn't make sense. And that's just because you have kind of only a portion of the protein there. And so maybe you want to go and explore and click down on some of these other ones and look and see. So here you can see that the structures contain positions 20 to 338. So it's missing the beginning portion of the protein. But if we go to PTM processing, we can see that actually the beginning part is just a transit peptide. That's what tells it to go to the mitochondria, and then it gets removed. So it's not even in the final structure. So basically, these are all pretty much full length. If you want an actual full length, well, here they have an AlphaFold predicted model. So you almost always are going to find an AlphaFold predicted model for a protein entry in UniProt. And you can see when you have an AlphaFold predicted model that there are going to be regions that are dark blue, which is going to be very high confidence in the prediction, and then regions that are like orange, which are very low. And so you can see that this region of the signal peptide is really orange, and it's like seems all disordered and stuff. Basically, that's the first part of the protein that's coming out. This is the N terminus. This comes out. It signals to the cells to take it to the mitochondria, and then it gets chopped off. So we don't really care about this usually because it gets chopped off. But if we want to know, well, what is it predicted to look like, we can see it here. And if we had a protein that didn't have a full structure predicted, a full structure solved, we could go and get a full structure this way with the AlphaFolds. However, if there's a structure that has the region that we're interested in, we often would prefer to go to the actually experimentally solved structure. So how do we know like which one to use? Well, it kind of depends because it's hard to know with just seeing these identifiers and the angstroms and things what these actually correspond to. So they could have various ligands bound, so various things bound to the protein. They could be the protein in different shapes and different conditions. So you can't really tell without, well, you can click on them to see a little bit more about each of these. And then you can also, if you wanted to find out even more information, you can go to the PDB. So you can conveniently link to the PDB entries from UniProt itself. So there are different PDB banks. So the protein data bank, they're like different servers. And so you can see there's like the European one, the RCSB, this is the Rutgers one, the US based one, the Japanese based one. And there's also like an AlphaFold database. So say I wanted to go and I wanted to find out more about this 2D FD structure. I'm just going to open this in a new tab. But basically what you can do is you can go and you can look at the structure and you can find out more about the experimental details of it, see what's in it. Maybe there are some other ligands bound. So this says like histidine and malate and stuff like that, which maybe you're interested in, maybe you're not. And you can go and you can look a little more at the structure, see what's in each of these different kind of chains. It's helpful to kind of like scroll down and you can see everything that's in here. And so it's going to tell you small molecules. It's going to tell you about, okay, well, these A, B, C, D chains are going to correspond to the protein. Maybe you only want one of them. Maybe you want them all. Doesn't matter for most things, but if you were to go and you wanted to go open this and say Pymol or Chimera X or whatever sort of visualization software you want to use, maybe you just want to do a single chain. But those are all just like technical details, and you can get a lot of information just by looking at the entry, and you can explore it in the web browser. But so you're in the PDB, and this is going to correspond to a structure of this protein. And again, you can go to the AlphaFold database, and you can find out more about the AlphaFold predicted structure. If you wanted to like go and get this in Pymol or something, well, you can download the PDB or the MMSIF. This is like you can open either of these types of files in most molecular viewing softwares. And so, yeah, so that would be your predicted model, and this would be your experimental model. And so if I look down here, you see AlphaFold predicted, and it's got this kind of weird identifier that starts with an AF, AlphaFold. And then these PDB codes are going to be 4 digits. And those were all for the experimentally determined models that have the PDB codes, these 4-letter codes, as opposed to just the predicted one, this AlphaFold model, which has this long weird code that starts with this AF for AlphaFold. And that will always be the full length protein. But it might not be accurate, especially in those orange areas. But that can be because the protein doesn't really have a set structure. And so, well, how can you predict something that is pretty random? Final word of caution, depending on the technique used, you might get kind of weird artifacts. So for example, so we know that the human mitochondrial malic dehydrogenase acts as a dimer, but you can see here it kind of looks like a tetramer. This is just an artifact of X-ray crystallography. You can see that these two kind of like dimers aren't actually interacting. They just happen to be kind of like in the same crystallographic space. And so don't get tripped off on that, but if you don't want to have to be dealing with like multiple subunits or something, you can always choose, you can probably choose a version that's going to be easier to work with. But you don't know what else is in the files, and maybe there's some other things about them that are different. And so you can go and you could explore the PDB entries to find out more. So these are just things that you might encounter when using the PDB or when using UniProt and all these tools. But hopefully things are more simple for the proteins that you want to look at.